It's my distinct pleasure to introduce the Chief Medical Officer at the Jackson Laboratory. Dr. Jens Rueter is a practicing oncologist and an expert in translational medicine. He's led groundbreaking research and application in cancer genomics and clinical initiatives to better personalized treatments for patients. I'm excited to hear about the work that Dr. Rueter and his team have been doing, really bringing precision medicine directly into patient care. So thank you, and Dr. Rueter, you're up.
All right. Thanks, Sam, for the introduction, kind words, and thanks to all of you for inviting me to speak here. Not sure what got into me to actually come up with this title. It's a little bit strange now that I look at it, and there is another version of it, which is this one. It's a bit more practical. So Sam was kind enough to introduce me. I am the Chief Medical Officer at the Jackson Laboratory, which is a little bit of an unusual position for a basic nonprofit, basic science/translational research organization. We are, for those of you who don't know, we are we have an NCI-designated basic science cancer center. It's actually quite interesting because it encompasses most of the faculty members at JAX. Actually, there's about, I believe, 60 to 70 faculty members on 2 different campuses, research campuses in Bar Harbor, Maine, and also in Farmington, Connecticut, and now also a small presence or a starting campus in Portland, actually right on the campus of the Roux Institute. And we largely, as I said, do basic and translational research, but we also have a clinically oriented program called the Maine Cancer Genomics Initiative, which will be the basis of my talk here. And I am, as you can tell from that title, or from Sam's introduction, also from what I just told you, I am not a specialist in AI and machine learning, but I had the distinct pleasure of actually, we started a nice partnership with Sam Scarpino and with Ben Giori here from Northeastern and Melanie Torrey, who's up at the Roux. To actually see how we can utilize AI in this clinical decision-making process. So what I will talk about here today is a couple of things. I'll start we have talked a lot about precision health, but not specifically about precision oncology. So I'll give a short introduction to what it actually is and what we actually mean by that. I will talk about something that is very near and dear to my heart that we have built over the last couple of years, a program of Genomic Tumor Boards. What are they? What are they doing? And then kind of circle back to AI. There will be a little bit of an intermezzo of AI in the middle between 1 and 2 here. But then talk a little bit about a project that we just recently started sort of conceptualizing and started working on that I think is very exciting and will hopefully be very impactful in this whole field. So when we think about precision oncology, what we mean is we think about the typical treatment of patients with lung cancer until about 15, 20 years ago. More or less everyone was kind of treated the same. If you had advanced cancer, metastatic cancer especially, you received some form of chemotherapy treatment. And that is still the case in some instances today. However, we have learned since that time that if we do a fairly extensive, what we call biomarker analysis on the DNA level, RNA level, protein level, with the DNA analysis being most advanced, that we can really individualize treatment and sort of parse out different treatment options for patients based on the markers that they have. Now, this seems like a great paradigm and fairly straightforward, right? So that's kind of the starting point to this. However, if you think about how this is then used in clinical practice, the reality looks like this. It doesn't look quite as clean as what I just showed you. It's a lot of different stuff, a lot of alphabet soup, quite frankly, on a piece of paper that a clinician will receive as part of, it's actually now standard of care to do this kind of testing, to do genomic testing, and then they will get this back, right? It's a long list of things, sort of terms that are not necessarily intuitive to a clinician, and it's not quite as straightforward as we would hope it to be, especially in an example like this, what I'm showing here. You can see that on the very left, on the left panel there, it talks about the so-called potentially actionable variants, right? What I showed you earlier is you look at a cancer and you get one specific marker that will identify one treatment. Well, oftentimes you have a list like this, and there might actually be 4 or 5 different treatments that are a possibility, and you would really like to know, well, how can I actually use this in clinical care. So how do oncologists actually make sense of this information, and how do they use it in clinical care? So just a couple of thoughts. They start searching. They start thinking about this. They start, trying to come up with a treatment plan based on this, which, is difficult because, as I just showed you, there we go. It's a little bit of alphabet soup, not always as well organized as in this particular bowl of soup here. And so really finding the right treatment is really a little bit like finding a needle in a haystack, right? Which is what then the clinicians are doing. So how can we help clinicians in making better decisions, and how can we help them find that needle in the haystack? So now, just full disclosure, will be the short intermezzo of AI that I want to bring up right now, which is this. Does anyone know what this symbolizes? Any chance? So this is a symbol of IBM Watson, right? IBM Watson. I'm sure many of you in the room have heard about IBM Watson. So the idea behind IBM Watson was, and I say was because my understanding is it's been shelved, or at least it never really took off the way it was intended to. It started in the early 2010s. IBM really invested a lot of resources into it, and the idea was you could feed a large, AI engine. You could basically train it with some clinical input, and it would then basically deliver answers to questions about the best treatment option for patients. And they did this for general oncology, and they did it for genomics as well. There was a specific platform called Watson for Genomics. And I actually remember this when it started sort of making an appearance and really kind of getting a little bit closer to the mainstream right as we were starting with the project that I'll talk about next, the Main Cancer Genomics Initiative, and it sounded too good to be true. Well, it turns out it was probably a little bit too good to be true in the sense that it didn't work. It didn't work in the sense that, most importantly, I'm sure there are technical reasons why it didn't work that many of you here understand much better than I do, but the main reason why it wasn't utilized and it didn't take off is because clinicians didn't trust it. And it produced some veritably false recommendations. And if you kind of Google this, you'll find some of these examples. So long story short, it didn't work. So that's not the solution, at least not at the moment. So what are the alternatives? Are there some other things that we can do to help clinicians sort of find that needle in the haystack and do the right thing? Well, back in 2015, 2016, the leadership at JAX, this is actually before I joined JAX, decided to do something about this problem. The problem of the complexity of genomic information, having access to testing, but then also interpreting the information, that really needed some support. So we received a large philanthropic donation from the Harrod Alfond Foundation in Maine. And we built this program called the Main Cancer Genomics Initiative, which really had the primary goal of essentially reducing disparities in care, because getting access to genomic testing and using it in clinical care was already becoming standard at large academic medical centers here in Boston and elsewhere, but certainly not in the community. And really, if you really boil it down to what we are trying to do, and this program is still ongoing now. We started, as I said, in 2016. We had received a second philanthropic grant from the Alfond Foundation in 2020, and this has really kind of allowed us to mature this program into something that I think we can make sustainable. But the entire goal of the program is to use advanced information from advanced genomic diagnostics and then turn them into better treatment options for patients. And the mainstay of the work that we do is that we developed this program of what we call genomic tumor boards. They're also sometimes called molecular tumor boards. And for the non-clinicians in the room, tumor boards are actually, in oncology, a very common forum to discuss complex cases that require multidisciplinary care from surgeons, from medical oncologists, radiation doctors, et cetera. Genomic tumor boards are very focused on the genomics of a patient's cancer and also sometimes the genetics that predispose them to cancer. So we developed this program as a non-clinical institution by partnering with all the medical oncology or oncology practices in the state of Maine. And we developed this sort of decentralized system of genomic tumor boards that would allow the clinicians to present their cases at a time that was convenient to them. That's important in clinical care, as we can imagine. And we called on one or two of these experts at a time, not all of them together, and the sort of faculty has shifted over time, to then actually opine on a specific genomic profile and to provide some input on what might be the best treatment option for the patient. So we did this, and you saw me waving my hands there, or my arms, in person for a while, and then with COVID, we shifted to virtual platforms, first WebEx and now Zoom. And we run these genomic tumor boards about once or twice a week for the state of Maine. For practices in Maine, then we also have now a national study that we're running through the SWOG Cancer Research Network that is actually a cluster randomized trial looking at the impact of these genomic tumor boards on patient outcomes. So what we do specifically doing a genomic tumor board is the following. We actually put a lot of preparation into preparing a case. You saw the test reports that I showed earlier. They come in just like that, right? It could be poorer quality. They could be just photocopied, scanned, faxed, whatever copies of a test report. So we're not talking about structured genomic data here. We're talking about really clinically relevant information. And then we put them into various formats that will help the clinicians better understand what they could potentially do about this. And we actually, one of the things that we do is to produce a table like this that is supposed to illustrate to a clinician, all right, you have a couple of treatment options, and this is sort of the highest level of evidence that supports using this. And from left to right, the level of evidence goes down. On the very left, you have FDA approval, and then you can see on the very right, you have phase 1 studies, so sometimes even only preclinical data. This is actually the actual table that we use in the Genomic Tumor Board to discuss the case that I showed just a few slides ago. And you can see there are a couple of things going on, but it's still quite a lot, right? So just looking at the evidence alone may not really be enough, because you still don't quite know what you're going to do with this patient. So as I said, we call on these experts to come in and help interpret the information. And this is actually a verbatim, more or less verbatim transcript of the expert who opined on this case. And you can see what's going on there. There's a couple of markers that we're addressing on the left-hand side. And he's kind of, slowly walking through the line of thinking, and the decision-making is sort of forming as you read from top to bottom on this, in this little narrative here. And you can see at the end, the punchline really is the last sentence here, which is, I would put my bet on immunotherapy, right? Because of this POL-E mutation that we also identified up here and the high TMB. So this is kind of what happens. I just wanted to illustrate that because it will be important in the next couple of slides as to why this is actually relevant. So prioritization of biomedical evidence is sort of the key in making this an effective program. So, and then we use this information to then produce a summary table that will focus on the one thing that is, or one or two things really, that we kind of landed on in this discussion, what the genomic marker is, and then sort of what the rationale is, or what the scientific evidence is behind it, ideally with a sort of a PMID number here so that the clinician can theoretically look up what the information is that we're presenting, which is important because clinicians like to know why certain recommendations are made. And they may actually use this as well for insurance, approval of medications, or they can use this to appeal. So it's quite an impactful document. So what we have created with this is sort of a system where the genomic tumor boards become sort of a central feature of this sort of feedback loop between the oncologist. The patients don't participate directly in this, although we've done this once, and that can be very interesting as well. But the patient interacts with the oncologist. There's a genomic test that's involved, which then is brought to the genomic tumor boards. There's peers and experts that kind of help the clinician come up with a plan based on precision medicine drug or a clinical trial that then gets delivered to the patient. We are looking at this as an experiential educational experience for the clinicians, and we also produce formal CME modules that the clinicians can refer to get some additional information. So it's been quite successful. We've been running this program since 2016. This is from 2 publications over the last couple of years, from the first 4 years of the program that we called MCGI 1.0. This shows sort of the enrollment of patients on the study that we had created. And you can see that especially rural areas, the kind of more red areas here means higher density of enrolled patients. rural areas enroll the majority of the patients. These gray areas might look disturbing. It just means there are not enough people in those areas to actually enable some population density measurements. And then we've also found that patients that actually go on targeted therapies actually have a better chance of being alive at 1 year. So there's an improvement in the so-called overall survival here. So those are kind of the impacts that we have achieved today. But the question that then comes up, how can we scale this? What we're doing right now is it's all manual. It's all manual work. It's manual work to organize these genomic tumor boards. It's manual work to take in these documents and produce a table like I just showed you. And it's manual work to then produce the summary. So this is, I think we started talking, Sam, maybe a few years ago already. This has been going on for a while. And we really were, it took us a while to really figure out what are the key elements that we need to kind of work on. Of course, first you have to sort of think about, well, what are the limitations? So the limitations to scaling are, first of all, there's only a limited pool of these experts that I just showed you. So you can't, scale beyond a certain number of meetings because people are busy and they're doing other things. It's a manual labor-intensive process. I already mentioned that. And there's actually also no standardized pool of knowledge. It's actually quite interesting since we work with a whole group of people that rarely actually come together and kind of exchange their opinions on how to treat patients based on a genomic profile. We've actually learned that some clinicians or some experts will interpret information differently than others. And so there's not currently a standard. They're all kind of learning from each other over time. But it's another issue if you're trying to kind of scale this and make it more broadly available is, you don't really, if you don't have a standardized pool of knowledge, that's kind of a problem. So how can AI then, since we're at an AI conference, how can AI help us to overcome these obstacles? Well, one of them, the whole process of the document intake and getting all the information in and making it presentable is certainly an area. It's one of these, I think I saw it on Jean's slide, the boring workflows. I mean, it's important, right? Because without workflows, we can't really exist. So I'm sure there's something that we're quite certain there's something that we can do there, and we've actually started working on this. And then the other one is we can also provide AI assistance to the expert, primarily in the beginning, right, to refine this expert decision making. So it's this whole concept of the expert in the loop or the human in the loop that we can really, I think, facilitate here. And if you look at this specifically, so how do we think this will actually work? I just want to walk you in a bit more detail through the workflow process at the current time. So we have input, what we call input data, right? We have patient clinical data, we have genomic diagnostic data that has to be put together in a package, then it's delivered to the GTB. There is a discussion at the GTB. There are queries during the GTB. Oh yeah, what was that study? I think I remember there was a publication 3 years ago. Those are the types of queries that we have to then also kind of channel. And then there's a summary that we, as I showed, that we then produce for every case that is discussed. So a couple of touch points here. So we've postulated that we can create a so-called workflow assistant that will help facilitate, again, the intake of these documents, and that will also help produce this summary in the end. And then secondly, the other part, though, and the one that's probably more complicated if you think about it, can we build an AI interface that will enable real-time access to relevant publications, to open clinical trials that are actually enrolling, so that that information can then be fed back into the GTB discussion in real time and not require any additional manual searches or delays, and also provide a consistent source of information, which is also, I think, highly relevant. So we've called this the GTB Decision Support Assistant. I say we, and it's really a collaboration that we just started, as I mentioned earlier, with Sam and Ben Giori and Melanie Torrey, and then Maine Health is also involved. We've worked for many years with the outcomes research group there to evaluate the impact. And we just actually put in a large NIH grant application for now, and we'll see how much this gets funded. It's obviously not a very, it's not sort of the mainstream, type of research questions that we have, but even if it doesn't get funded, I think we'll find ways to continue this work, because it's already, some of the preliminary work that we've done already shows that we can indeed, especially on the workflow assistant side, make quite some impact. So I think it's important. I asked the question earlier about evaluation. I think we have to have an evaluation in there. about, what are the actual savings, how reliable is this? We're talking about, again, clinical decisions. And I think it's important that the information is correct, and not only correct most of the time, but ideally almost all the time or all of the time. And then also the trust question that came up earlier. I think it's really important to evaluate trust of the people that are currently producing the workflow documents, but also trust of the experts as they're utilizing information. And then, of course, the most important question, I think, can we use any of the data points that we collect in that process to improve the output and make it better over time? Just, again, this project just started. I'd be happy to come back in a few years and report on what we found and what we've been able to do. But for right now, the conclusions are that clinical decision-making is very complex. We know that we can implement something at a regional level, i.e. on a state-based level with appropriate funding. Right now, we can't just simply replace human experts. Maybe we never will be able to. But we can definitely work on sort of these support systems that will help clinicians and experts make better decisions and deliver, a more streamlined process and enable scaling. I think that's the most important part. That's the end of my talk, and I don't know if I have time for questions, but I'm happy to answer any questions.
Thank you for a fantastic talk. 2 comments and perhaps a suggestion. Just wanted to point it out that in HIV medicine, this model of your tumor boards was used for multi-resistant HIV virus, bringing virologists and pharmacologists and the clinician together around the table, and proved to be extremely efficacious. We did not have AI in those days. My question/comment is, one of my understanding was that the IBM experiment failed also because it was perhaps collecting what was more was better philosophy, collecting too much data from literature, etc., rather than focusing on a more focused data. Can you share your thoughts on that?
That's a great question. That could be part of the problem. I think, and again, as I said, I'm not an expert on why Watson didn't work out in the end. I would argue that it is indeed true. We actually use a knowledge base right now that was originally developed at JAX called the JAX Clinical Knowledge Base. There we can actually filter, the evidence that we're looking for. I think if you're searching, it's always, I think it would be hard to know upfront as to what you would focus on, right? Because what's clearly also we've identified over the years that things that seem more relevant in clinical decision making, for example, are things that are fairly recent. So what I mean by that is if you had a phase 1 study with a certain approach that was published, 10 years ago, and nothing else ever became of it. We actually go through this exercise. If we find a reference like this, we actually try to find an internet trail of what happened. What happened to the drug, what happened to the approach, and sometimes you can find it and sometimes you can't. We've kind of come to the conclusion that probably things that happened 10 years ago and didn't go any further are probably not relevant, right? So focusing on things that are more current for the most recent drugs. However, if it's sometimes, that's all you have are these older references. So it's not easy a priori to decide what's valuable and what is not valuable.